Hello, Biology 1 student. This lesson is on Competency 1, Inquiry, or the scientific methods. I have included your Competency 1 standard and proficient PLDs for informational purposes only. This slide shows your assessment verbs and scientific vocabulary associated with Competency 1. Before we get started, please download and complete the four-square vocabulary sheet for assessment verbs and scientific terms that are unfamiliar to you. This lesson is designed to review the Competency 1 biology content you learned in your classroom. You will be asked to apply the component of scientific processes and methods in scientific investigations. You will be asked to analyze graphs. You will analyze procedures, data, and conclusion to determine the scientific validity of research. Science is an organized way of gathering and using evidence to learn about the natural world. Scientific methods are organized series or events used to answer questions and solve problems. Like all science, biology is a process of inquiry and begins with careful and systematic observations. These are the steps of the scientific methods. Your steps and processes: observing, asking questions, forming hypotheses, designing controlled experiments, collecting and analyzing data, drawing conclusion. Observation is the act of noticing and describing events or process in a careful, orderly way. Observation leads to question. Here's an example of an observation in real life. You got in your car to drive to school. You turned the ignition key, but the car wouldn't start. This is an observation. You ask yourself, what is wrong with my car? This is a question or a problem. This is a real life example, but scientists use these same types of observation and questions in their research. A hypothesis is a scientific explanation for a set of observations that can be tested in ways that support or reject it. My battery is dead. This is my first hypothesis. I am out of oil. This is my second hypothesis. I am out of gas. This is my third hypothesis. Controlled experiments are designed to investigate questions or problems. Whenever possible, a hypothesis should be tested by an experiment in which only one factor or variable is changed. Let me repeat that because it's very important. Only one factor or variable should be changed in a controlled experiment. These are my experiments for my car. I turned on the headlights and radio. This was my experiment for hypothesis 1. I used a dipstick to check the oil. This was my experiment for hypothesis 2. I looked at the fuel gauge to determine how much, if any, gas was in my tank. This was my experiment for hypothesis 3. Controlled experiments. Independent variable. The variable that is deliberately changed is called the independent variable, also called the manipulated variable. The independent variable is the tested variable, and it might affect the outcome of the experiment. Controlled experiments, dependent variable. The variable that is observed and that changes in response to the independent variable is called the dependent variable, also called the responding variable. The dependent variable results from or depends on changes to the independent variable. These are the steps of my controlled experiment. A typical experiment is divided into control and experimental groups. A control group is used for comparison. It is exposed to the same conditions except the independent variable. An experimental group is exposed to the factor being tested. Again, your independent variable. As scientists test their hypotheses, they make detailed records of experimental observation, gathering information called data. Data collected as numbers are called quantitative data. For example, if I ask you for quantitative data of your classroom, you might tell me something about the width or the length of the room. You will use number for this quantitative data analysis. Data collected as descriptive characteristics are called qualitative data. If I ask you for qualitative data about your classroom, you could tell me something about the color of the classroom. Again, describing quantitative data about the classroom. Let's look at the data analysis for my car. I was able to turn on my headlights and radio. This is experiment 1. The dipstick indicated my oil was at the appropriate level. Experiment 2. My fuel gauge indicated I had no gas in my tank. Experiment 3. Conclusion or theory. Scientists use experimental data as evidence to support, refute, or revise the hypothesis being tested and draw valid conclusions. When enough evidence from a wide range of observation and experimental results supports a hypothesis, scientists call the proposed explanation a theory. My conclusion for my car, based on my experiment and recording my data, I am out of gas. Here's another example of an experiment. Wilma Flintstone and Betty Rubble are best friends. However, both have very competitive personalities. Wilma observed that Betty roses were taller and contained more blooms than her roses. She noticed that Betty used a new fertilizer, rock. This observation leads to a question. Does rock fertilizer increase the height and number of blooms in knockout roses? Hypothesis. Rock fertilizer would increase the height and number of blooms in knockout roses. This is Wilma's controlled experiment. Wilma made a trip to the local Bedrock Garden Center. She purchased 15 knockout rose bushes of similar age and height. In her backyard, she selected 3 plots. All of the plots were similar in size, type of soil, and source of light. Wilma planted 5 plants in plot A, 5 plants in plot B, and 5 plants in plot C. All plots received the same amount of sunlight and water each day for 4 weeks. Plot A received rock fertilizer. Plot B received Wilma's fertilizer. Plot C received no fertilizer. Let's examine Wilma's data. In week 1, all of the plants in each plot were 65 centimeters in height and had 10 blooms. In week 2, the height of plants in plot A increased from 65 centimeters to 70 centimeters. The height of plants in plot B increased from 65 centimeters to 66 centimeters. The height of plants in plot C did not change. In plot A, the bloom number increased from 10 to 16. In plot B, the bloom number increased from 10 to 12. In plot C, the bloom number did not change. Let's look at week 3. The height of the plants in plot A increased from 70 centimeters to 78 centimeters. The height of plants in plot B increased from 66 centimeters to 67 centimeters. The height of plants in plot C increased from 65 centimeters to 66 centimeters. In plot A, the bloom number increased from 16 to 24. In plot B, the bloom number did not change. In plot C, the bloom number increased from 10 to 11. Let's look at week 4. The height of the plants in plot A increased from 78 centimeters to 92 centimeters. The height of plants in plot B increased from 67 centimeters to 70 centimeters. The height of plants in plot C increased from 65 centimeters to 67 centimeters. In plot A, the bloom number increased from 24 to 31. In plot B, the bloom number increased from 12 to 15. In plot C, the bloom number increased from 11 to 12. And here's a graphic analysis of the data you found on the previous slide. This is a bar graph. Drawing conclusions. After careful analysis of data, Wilma concluded that knockout roses grown with rock fertilizer produce taller bushes with more blooms. Let's practice again. Derrick Rose and Dwayne Wade want to impress the fans with longer hang time during the next NBA slam dunk contest. Reebok advertises a new shoe that would increase hang time by more than 50%. Derrick wears the new shoes and finds that his hang time increases from 4.1 to 4.8 seconds. Dwayne does not wear the new shoes and his hang time remains at 4.2 seconds. Your questions? What is the problem? What is your hypothesis? What is the control group? What is the experimental group? What is the independent variable? What is the dependent variable? What are your conclusions? Let's take a moment for you to answer these questions based on the scenario. Your answers should be very similar to the answer you see here. Problem: will the new Reebok shoes increase hang time by more than 50% during dunking? Hypothesis: the new Reebok shoes will increase hang time by more than 50%. Control group: Dwayne not wearing the new shoes. Experimental group: Derrick Rose wearing the new shoes. Your independent variable, remember this is the variable that you are changing. Wearing the new shoes. Dependent variable, remember this is your responding variable. In this case, the length of hang time is your dependent variable. The conclusion of the experiment shows the new Reebok shoes did not increase hang time by 50%. Let's practice again. Madison has a big mustard stain on her new expensive t-shirt. She is afraid to tell her mother and decides to clean the t-shirt herself. She seeks advice from her chemistry teacher. He suggests she treats the stain with something before washing it. The biology teacher suggests she pre-treat the stain with hairspray. The history teacher suggests she pre-treat the stain with vinegar. The physics teacher suggests she pre-treat the stain with salt water. Madison thinks that no pre-treatment is required. Your task: design a controlled experiment to solve Madison's dilemma. Let's take a few moments for you to do that. Your answers should look something very similar to the answers here. Problem: what effect does pre-treatment have on removing mustard stains from cotton fabrics? Hypothesis: pre-treatment would have no effect on removing mustard stains from cotton fabrics. Controlled experiment: Madison cut 4 pieces of cloth, same size, from an old cotton t-shirt and stained each with a tablespoon of mustard. She pre-treats cloth 1 for 2 hours with vinegar. She pre-treats cloth 2 for 2 hours with salt water. She pre-treats cloth 3 for 2 hours with hairspray. She did not pre-treat cloth 4 at all. She placed all 4 cloths in the washer on a regular hot water cycle. The control group, cloth 4 with no pre-treatment. Remember, the control group is not exposed to the variable. It is a comparison. Experimental group, cloth 1 with vinegar, cloth 2 with salt water, and cloth 3 with hairspray. Your independent variable, the variable that you're changing or manipulating, would be pre-treatment of mustard stain. Your dependent variable is the removal of the mustard stains. And here's the data collected from Madison's experiment. If you notice that cloth 1, that is the one pre-treated with vinegar, there was complete stain removal. Cloth 2, pre-treated with salt water, no stain removal. Cloth 3, hairspray, very little stain removal. Cloth 4, no pre-treatment and no stain removal. So, conclusion of Madison's experiment, pre-treatment of mustard stains with vinegar will remove the stain from cotton fabric. Graphing is a very important part of the scientific methods. All graphs should have a title. This shows what the graph is about. Reading the title should give the reader an idea about the graph. It should be a concise statement placed above the graph. The independent variable, this is the variable part of the experiment that is changed, that can be controlled or manipulated by the experimenter. This variable is placed on the horizontal or x-axis. The dependent variable, this is the variable that is directly affected by the independent variable. Remember earlier we said it's your responding variable. It is the result of what happens because of the independent variable. This variable is placed on the y or vertical axis. In constructing a graph, one needs to know where to plot the points representing the data. In order to do this, a scale must be employed that will include all the data points. Each block should have a consistent amount or increment on a particular axis. Here's an example of a bar graph. The title of the graph is Peaches Picked in an Orchard. And we have our years placed on our x-axis because that is the changing variable. And the number of kilograms of peaches collected during each year is placed on the y-axis because it is the responding variable. Again, your independent variable is placed on the x-axis, which is horizontal, and your dependent variable is placed on the y-axis, which is your vertical axis. Let's practice looking at data analysis. Chemists working for a pharmaceutical company carried out an experiment in which they measured the amount of time it takes for different amounts of an acid to neutralize 1 milliliter of hydrochloric acid. The results of their experiment are shown in the graph below. Let's take a moment to look at the graph. Make sure you study what's on the x-axis as well as the y-axis, your independent and dependent variables. These are your questions. What is the relationship between the amount of an acid and the length of time to neutralize the acid? What are the independent and dependent variables in this experiment? Here are your answers. Large amounts of antacids will neutralize acids in a shorter time span. Your independent variable will be the amount of antacids. Your dependent variable, acid neutralization time, because it is going to respond to the antacid or the amount of the antacid. Here's another data analysis practice. A group of students studied the effects of light intensity on the rate of cell process in LDL plants. The students exposed LDL plants to different light intensities. A gas was produced by the cell process. The amount of this gas was measured. The rate of the cell process was determined by the amount of gas produced. A graph of the students' measurement is shown below. Let's take a moment to look at the graph. Which of these is the independent variable in this experiment? If you look at the graph, you will see your X and Y axes are labeled. So here are your answer choices. A, rate of cell process. B, volume of gas. C, size of LDL plant. D, intensity of light. In this graph, we know that the x-axis is labeled, and we know that the x-axis contains the independent variable, so D is your correct answer choice. Here's another practice problem. According to the graph, how many mice will be born in week 5 if the trend continues? So let's take a moment to look at the trend. If you notice in week 1, we see the number of mice will be 10. In week 2, 20. Week 3, 40. Week 4, 80. And by analyzing this and looking at the data, we can see that each week the number doubles. So we can say that the number of mice born in week 5 should be 160. So D is the correct answer because it will double 80 and also follow the previous week's trend. Here's another practice scenario. A zoologist set up an experiment to study 9 mice. On day 1 of the study, the mice were measured for a variety of characteristics. Then the mice were put into cages and kept under the following conditions. Cage 1, he placed mice 1, 2, and 3, 12 hours of light per day, 6 ounces of food per day, a temperature of 72 degrees Fahrenheit. There was a wheel for exercise, and the cage was 4x6. In cage 2, he placed 4, 5, 6, and 7. There were 12 hours of light per day, 6 ounces of food 1 time each day. The temperature was 86. There was a wheel for exercise, and the cage was 4x6. In case 3, he placed mice 8 and 9, 12 hours of light per day, 6 ounces of food 1 time each day. The temperature was 60. There was a wheel for exercise, and the cage was 4x6. These are your questions. What is the variable being studied? How do you know this? What flaw in the design of the experiment can you find, if any? Let's take a moment to look at the chart again and answer these 2 questions. Here are your answers. What is the variable being studied? How do you know this? The temperature is the variable being studied. It is the independent variable because it is the only thing that is changed during the experiment. All the other variables are kept the same. Therefore, the temperature is the variable being studied, your independent variable. What flaw in the design of the experiment can you find, if any? The zoologist should have used the same number of mice in each cage. If you notice, he placed different number of mice in each cage when they should have been the same number. Here's another scenario. Is the scenario below a good or flawed experiment? How can you improve it? Richard wanted to test which brand of car wax was most effective. He tested 4 brands of wax. He cleaned the hood of his car and removed the old wax. He measured 4 equal sections on the hood of the car. Each brand of wax was used to cover a section. An equal amount of wax, the same cloth, and equal buffing were used. He collected data by developing a rating scale of 1 to 3. 1, very shiny. 2, shiny. and 3 dull. Here are your answers. Is the scenario below a good or flawed experiment? It is a flawed experiment because he introduced too many variables. How can you improve it? Richard should not have used the same cloth to spread each type of wax. He should have used the same type of clean cloth for each wax. Let's practice. Again, 4 teams of students independently tested the effects of temperature on the gill's movement of goldfish. The results from their tests are shown in the table below. Students from other teams questioned the results of Team 3. Which of these should the students on Team 3 do to validate their results? A. Use data from one of the other teams for the counts taken at 15 degrees Celsius. B. Repeat the experiment at 15 degrees Celsius using the same set of goldfish. C. Using containers made of different materials to test the goldfish again. D. Repeat the experiment at 15 degrees using larger goldfish. Let's look at the answer choices again. A. Use data from one of the other teams for the counts taken at 15 degrees C. B. Repeat the experiment at 15 degrees C using the same set of goldfish. C. Use containers made of different materials to test the goldfish again. D. Repeat the experiment at 15 degrees Celsius using larger goldfish. We can eliminate A, C, and D because we are introducing different variables. Remember, a good experiment tests one variable. All right, let's review. Read the experiment scenario below. After studying about recycling, members of John's biology class investigated the effects of various recycled products on plant growth. John's lab group compared the effect of different age grass compost on bean plants. Because decomposition is necessary for the release of nutrients, the group hypothesized that older grass would produce taller bean plants. 3 flats of bean plants were planted with 25 plants in each flat. The plants were fertilized as follows. Flat A, 450 grams of 3-month-old compost. Flat B, 450 grams of 6-month-old compost. And flat C, no compost. The compost was made of the same materials and the plants received the same amount of sunlight and water each day. The plants grew for 30 days. After 30 days, the group recorded the height of each plant in centimeters. Plants in flat A increased 10 centimeters in height. Plants in flat B increased 9.5 centimeters in height. Plants in flat C increased 3 centimeters in height. Here are your questions. What is the problem being studied? What is your hypothesis? What is the control group? What is the experimental group? What is the independent variable? What is the dependent variable? What data would you place on the X and Y axis? What are the conclusions? Let's take a moment to analyze the experiment and answer the questions. Your answers should look very similar to the answers you see here. Problem: does using older grass compost as fertilizer produce taller plants? Hypothesis: older compost grass produces taller plants. Control group, flat C. Experimental groups, flats A and B. Conclusion: the age of grass compost has no effect on increasing the height of plants. Independent variable, age of the compost grass. Dependent variable, height of plants. Your x-axis will contain your age of the compost grass because that is your variable that is being changed. And y-axis will be the height of a plant because that is your respondent variable or your dependent variable.